Hi, welcome to Caltech. A few housekeeping notes before we start. First, we ask that you silence your cell phones and look around to note the nearest exit in the event of an emergency. After the talk, we're going to have a question and answer session, and those with questions will be invited to mics in the aisles. After the program concludes, please remain in your seats because we're having a special raffle, after which we'll release you by school. So please wait for your group to be called before you file out. With that, my name is Kitty Cahalan. I'm the Assistant Director for Educational Outreach at Caltech Center for Teaching, Learning, and Outreach. CTLO is dedicated to building Caltech connections with students in Pasadena, Greater Los Angeles, and across the country. Alongside colleagues in Caltech public events, we work to ignite curiosity, mentor future scientists, and offer you all a glimpse into the exciting science and engineering that happens on our campus. We have a teaching program at a local elementary school, attend science nights at schools near Caltech, create summer research opportunities for high schoolers, and we help identify great speakers for the Science Journeys program. That's why you're all here today. Caltech has been presenting science talks like this one for more than 30 years. The science journey you're about to be part of will weave together cutting-edge research with the life story of one of our up-and-coming scientists. We're happy to have you here as our live audience. Our speaker today, Dr. David Delgadillo, is a postdoctoral fellow in the laboratory of Professor Hosea Nelson. David grew up in the Los Angeles area and attended UC Merced, where he majored in human biology with a minor in chemistry and earned his bachelor's degree while conducting research in the laboratories of Professors David Britt and Ryan Baxter. He continued his studies at UC Santa Cruz, where he worked under Professor John McMillan, earning his PhD in organic chemistry in 2022. We're very excited to have you hear his talk, and so without further ado, come on out, David.
So my name's David. Nice to see you all. Thank you all for coming out. Today I'm going to talk to you a little bit about my journey to Caltech, right? How a lot of my mentors kind of shaped my path here and changed my trajectory throughout my entire life. And then towards the end, we're going to touch a little bit on the science that I do here at Caltech. But before we begin, I'd like to talk to you all about the life cycle of a mushroom. I'm sure some of you have been taught the life cycle of a plant, but not many of us learn about the life cycles of mushrooms. All right, so many of us have walked around our neighborhood or in the forest, and we've seen this peculiar-looking little thing that we call a mushroom. And we typically think of it like a tree, but there's some subtle differences, right? If you flip the mushroom upside down, you'll see some gills, right? And they look like stripes, but these gills are responsible for dispensing these things called spores, right? And these spores are synonymous to seeds, like that of a tree, right? And these spores also go through a germination phase that eventually become hyphae, and we can think of this hyphae as the sprout of these seeds, right? The beginning of the tree. And then these hyphae eventually come together and they form a network of filaments that we call a mycelial network, right? And this is representative of the tree that we see in the forest, right? So most of a plant's life is above ground and most of a mushroom's life is below ground. And eventually those mycelial networks come together, they form a hyphal knot, and then buds a mushroom, right? And that drawing's pretty bad, so I kind of took a little video from the internet to show you what it looks like in real life, right? So here you'll see a little mushroom, and in the background you'll see some dust, right? Those are the spores dropping out of the mushroom beneath the gills. And here we're using an optical microscope to magnify deep into a pile of spores, and you can kind of see them floating around. They look similar to seeds, right? And eventually, they'll start to form hyphae, right? Little nodes or sprouting networks. And eventually, you'll come together and see some mycelium. And some of you might have seen this, like on a piece of bread, right? Or an orange that was left out for too long. So that should be familiar. Mycelium doesn't only grow on solid objects, it could also grow in water, so liquid media. But eventually, you can get a full mycelial cake. And over here, we have an actual setup of a mushroom kit that I think somebody's gonna raffle off later on. But you can see there's mycelium colonizing this entire substrate. And under the right conditions, right humidity and the right air exchange, you can start to see that mycelium form the hyphal knots and produce the fruiting body, which is the mushroom. And I have a cool time-lapse video to show you that happening in real time, or not in real time, but you'll see this mycelium taking over and colonizing substrate, and eventually it'll grow. So if you win this mushroom kit, you'll get to see this happen in your own house. Cool. So why am I talking to you about mushrooms and the mushroom cycle, right? I think I like this analogy because mycelium, right, can pretty much grow on almost any substrate, right? You can grow it on popcorn seeds, rice, an orange peel, water, whatever, right? So it's very resilient. And mycelium is also very good at forming networks, right? They can socialize with each other and exchange nutrients and information. And most importantly, all of it happens underground, right? We never see any of that. And so when I think of everybody else that I meet, right, I think of the mycelial network that happens beneath them, right? Something that I don't see. So today I'm gonna talk to you about my own mycelial network, right? The people and that have provided me nutrients and have helped me navigate my life. So here, I'm gonna talk to you about my mycelial network. It starts off with my two parents, Martha and Alfredo, who immigrated to Los Angeles from this small little town in Mexico called Concepcion de Buenos Aires in the state of Jalisco. And my journey started because of them, and I was actually born here in Bellflower, and then moved over to Hacienda Heights, where I attended a public school, public elementary school called Los Molinos, right? And around this time, I started thinking, or I guess my teacher started asking me, what do you want to be when you grow up, right? I really didn't know, but I asked my mom to pull up some of my old school projects. And it turns out I wanted to be an astronaut when I was a little kid, right? I was obsessed with stars. It doesn't look like it, but that foil is supposed to be my astronaut suit. And there's these tiny little yellow stars there, but I was really obsessed with the sky. I was really interested in it. Would always beg to go to the Griffith Observatory. If you haven't gone, you should go. It's one of my favorite places in the world, probably. And so these two people, my parents, have really nurtured and kind of fostered my interest in a lot of weird things like space or bugs or whatever science projects or experiments I did as a kid. And so thank them for supporting me and those curiosities. But, you know, growing up in an immigrant household, you're not told that being an astronaut's one of the best careers to choose. You're always told, you know, you should either be a lawyer, an engineer, or a doctor. And those are great careers, don't get me wrong. But I wanted to be an astronaut. So how did I get from wanting to be an astronaut to where I am today? I think a lot of that change in trajectory comes from the time that I spent in Mexico on my dad's father's farm, right? He ran a dairy farm. And when we would go there, this thing is a large, untouched land full of cattle, horses, donkeys, cattle dogs, all kinds of plants, all kinds of streams running through it. And it was probably a kid's dream, right? There was a level of exploration and freedom that I couldn't get here in Los Angeles. And while we were there, right, spent a lot of time. Here's a photo of my mom and I next to a little baby calf, or my dad and my little brother looking over at some of the cows as they're getting milked. But throughout this time that I spent there, right, I got to see my grandpa and my dad and the other work hands take care of these cattle, right? They spent a lot of time, protecting them by using antibiotics, right? They would always have these syringes, and I'd be wondering why are they sticking needles in these cows, or how did a cow who cut themselves on the barbed wire get healthy after a while, right? And there was always these antibiotic ointments that were readily available, and I think from that exposure, I really wanted to go into the healthcare services. I don't know, my mom could correct me if I'm wrong, but I think early on, maybe 5th, 6th grade, I decided I wanted to be a doctor. And so that was kind of my dead set career path. So after elementary school, I went to Newton Middle School, another public school. And it was around here that I kind of started to lose focus of school and what I wanted to do. I wasn't as keen to being the nerdy kid that I actually was, and I decided to pick up skateboarding. There's a photo of me up there in the 8th grade, so some of you might be in the 8th grade, and I would carry around my skateboard everywhere. All I cared about was skateboarding. But I eventually ended up going to Los Altos High School, right? And here I started to care about school even less, right? I was a lot more in tune with my friends and what I wanted to do with them, and started developing a lot of behavioral issues, right? I was probably the principal's best friend, but not in a good way. I was in and out of her office very frequently, and I think a lot of those behavior issues kind of made me sink down lower and really started to write myself off from going to school, right, and being good at school. But it wasn't until I took a class with this teacher named Winnie Gilbert, right, who saw me, I guess the first interaction that I realized she cared was after an exam. She came over to me and she was like, every time I see you take a quiz or an exam, you're not breathing. She's like, just remember to breathe, right? Your brain needs oxygen in order to function. And I still remember that till this day whenever I'm feeling stressed or nervous like I am now, I'm just breathe. And that was a budding relationship that carried out throughout my high school career, where I would rely on her a lot to not just teach me math or help me get through difficult exams, but to ask her about life, right? She was the one that asked me, like, are you applying to college? Are you gonna go to college? And eventually, I did apply. I did all my applications in one day, didn't revise any of it and just submitted it. Surprisingly, it got rejected by most of those schools, right? But I had a GPA that was good enough to go to a UC, and so I ended up getting an offer from UC Merced, which is one of the newer UCs, even though I didn't apply, but I guess the UC system has that rule in place. If you have the certain academic credentials, right, they'll have a space for you at one of their campuses. And so I remember going to her classroom and being like, should I go here or should I go to San Francisco State? And she knew that I wanted to kind of go into the healthcare industry. And I remember her telling me, you should go to the UC school, right? The UC system will give you the resources to put you at a competitive edge to go to medical school, right? So I decided, all right, I'm gonna listen to Ms. Gilbert. And that's pretty much where my academic career really changed. The school had just graduated their first class of college students in 2013, right? Or 2009, and so by the time I joined, there had only been 5 other classes there. And so it was a very new school, very small class sizes, but that seemed to be perfect for me. It helped me really focus. And I remember as I decided a major, I chose to do human biology, but when you're on the pre-med track, you have to take organic chemistry. And I remember a couple of my friends were in the honors organic chemistry class, and I wanted to also be part of that. So I went to this teacher, or this professor that had just started there, who was teaching the honors course, and I asked him, can I please join this course? It was only offered to chemistry majors. And he said, yes, I'll give you the chance, but only if you really, really put in the hard work. And so I did. I ended up having probably the best grade in the class, and he ended up asking me to work in his lab. And that's where really all of my organic chemistry research and interests started off. We started doing a lot of synthetic organic chemistry, so we're making molecules, and we'll get to that later. But But they also mentored me a lot on how to navigate higher education, right? I didn't have a lot of guidance from people around me on how to navigate those different courses. And so they taught me, the importance of applying for summer research internships and fellowships, the importance of actually conducting meaningful research and how to interrogate different research papers that you read on the internet, how to choose which ones are important. Shortly after working in that lab, right, I got some fellowships and I got the opportunity to work at UC Davis during the summer. And that's where I met Lizzie Tao, which was a graduate student at the time and then transitioned into a postdoc. But when we were making these molecules in Merced, right, we were working with what we call radical electrons, right? Chemistry works, or you're taught that it works in 2 electron mechanisms, but then what we were doing in the lab was 1 electron. And I, couldn't imagine that being possible, so I wanted to see how that worked, right? And so when I applied for some of the labs to work in Lizzie's lab in David Britt's group, they were actually studying and using ways to analyze and visualize those single electron mechanisms. And so I went off to work with her, but besides the really cool research and impactful research, she taught me a lot about life, right? We went through something that a lot of people usually don't go through, but we learned that science isn't everything, right? You're gonna fail a lot of the time. Most of the time you fail. Every experiment you do, there's a chance you're gonna fail, but those failures shouldn't define you. And so she and I really grew together and we maintained contact, and now she's a very prominent scientist at Southern University in China. But after summer research internships, I was able to finally graduate with a bachelor's in science in human biology, and this is probably one of the proudest days of my life. My whole family showed out, right? But after that, I ended up applying to graduate school, right? So I went from human biology, now trying to study chemistry, and I ended up going to UC Santa Cruz, where I met another mentor, John McMillan, who taught me everything that I know about what you're gonna hear about in just a few moments, right? He taught me how to look through nature and extract molecules and how to characterize them, how to visualize them, and how to know exactly what their properties are, right? I eventually got my doctorate, and then while I was at UC Santa Cruz, right, just like how mycelium tend to reach out and form networks to exchange information, when you're doing science, it's important to go to conferences, right, to exchange some of that information, and you might meet somebody, right, that might be your next mentor. At one of these conferences, I met this guy that might be familiar to some of you, named Hosea Nelson, right, and he had just published a really impactful paper utilizing electron diffraction in a way to characterize small molecules. Although the technique was known to be used on proteins, it was his group and his collaborators that came together and were the first to apply it to small molecules, right? And when we talked at this conference, he heard the science I was doing, I heard the science he was doing, and immediately we're like, let's do this together, right? And so he actually let me join his group through Zoom, because it was during the pandemic, and I would sit there on all of his group meetings for about a year. And eventually, he asked me to meet with him, and he was like, are you just a weirdo that's obsessed with my science, or do you want to come work with me? And I said, of course, I would love to come work with you, and that's how I ended up here at Caltech. So here's my mycelial network, right? This is my map, and I think if there's anything I want you all to take away from this is this quote that's up here by Oprah Winfrey. All of these people pictured above not only allowed me to see hope in my own self and my own future, but really inspired me to achieve more and want more. And now we're going to go away from a little bit of my story and talk about why mycelium, why is that the theme of this talk. Have any of you played this video game? Yeah? All right. Or if you haven't played the video game, you might have seen the TV show, The Last of Us. All right? So the premise of this show, right, is that there is a parasitic fungus called cordyceps, and it's supposed to take over the human body and turn your brain into a zombie. And although it seems far out, this is actually based in reality. Right? There is a fungus called cordyceps that is known to do this with insects. Right? It'll go and infect their nervous system, and then it'll turn them into zombies, lead their body into the perfect environment to where it can then produce the mushroom, right? And then drop spores and continue the life cycle. And although it's pretty far-fetched to think that that same thing will cross over into humans, it still is kind of scary, right? And I like to bring this analogy up because we just all experienced a pandemic with the coronavirus, right? And there's an impending antibacterial resistance pandemic, right, where we don't have a lot of antibiotics to our arsenal. There's even less antifungals, right? So even though this is far-fetched, right, if in the case that were to happen, do we have enough chemicals in our inventory to be antifungal, to help us combat against those fungal infections? The answer is we have some, but we need more, right? There's already been some fungal infections that have been shown to be resistant to all 4 classes of the molecules that we know to be effective, and so we're starting to get to that point where we're running out. So, mycelium, turns out, fungus is probably the best way to fight a fungal infection. And that is because there's a long history of these medicines, or these organisms producing medicines. Here you'll see a common piece of bread mold, some dirt. You might not see the fungus or the mycelium, but it's there. Or in plants, right? You can see them being infected by these fungi. Or you can see these funky, gooey-looking things out in the forest. But all of these have been known to produce really important molecules, important medicines, right? Such as penicillin, which is something that most of us in this room have probably taken to help some type of bacterial infection. Or lovastatin, which is super important to help regulate cholesterol. Ergotamine is super important for helping relieve cluster headache pain. And cyclosporin A is an immunosuppressive agent, right? So if somebody's getting an organ transplant, in order to help the body not reject the new organ, you would want an immunosuppressant agent, such as cyclosporin A, to help you take in the new organ. And these compounds right here are all small molecules. So throughout the remainder of the talk, I'll mention them as natural products or small molecules. And the truth is, they're not just important from fungus, right? They come from all types of plants. And the history of looking for medicine in nature is as old as time, right? One of my favorite examples is that of the willow tree. A lot of indigenous people used to chew on the bark of a willow tree if they had a headache or a fever. And that's because there was a molecule called salicylic acid in there that was responsible for anti-inflammatory properties. Another example is that of the mint leaf, right? It's especially relevant to me because I remember going through one of my grandparents' cabinets, and I think even my dad might have some of this, but rubbing alcohol, soaking with different herbs, right? One of those could be mint, right? And so the thing was always like, oh, you can only use a certain herb for a certain ailment. Like if you got a cut, you use this herb. If you got a burn, you use that herb. Turns out there's a compound called carviol that is also anti-inflammatory like salicylic acid, but it's also got some subtle antibacterial properties. So if you're trying to clean a wound, it all of a sudden starts to make sense. Another thing that's pretty relevant is the aloe vera plant, right? I don't know, this is all kind of based on home remedies, right? But my parents have an aloe vera plant outside our front yard, and every time my mom accidentally touches a hot pan and gets a burn, she screams, "David, go run and get me some aloe vera." And the process goes, you grab one of these leaves, and then you end up cutting the leaf, and you get the little gelatinous portion on there, and you apply it to the burn, right? And so as a kid, and even till now, I've always wondered, like, does this actually even do anything? Is there something important in here? After doing some research, there's actually a molecule called asamanin that stimulates the immune response, right? And there's also certain levels of salicylic acid in there. So if you do get a burn and you apply this gelatinous thing, it would make sense that you're relieving some of that pain and stimulating an immune response. And so there's a lot of these examples, but that's just to say there's molecules out there in nature that can benefit us all. And then these molecules and knowing their structure is super important, right? Because if you look at the original natural product salicylic acid, if you've ever taken aspirin, these molecules look almost identical, right? And so solving the structure and knowing what they look like will help scientists change them to perfect their properties, right? To make them more effective, to relieve pain at a higher degree. And so we can make changes like this where we swap the hydroxyl group for an acetate group. And so the last example here, right, is the example of the cinchona plant, right? A lot of indigenous populations also used to use this to relieve a lot of fevers, right? And it wasn't until several years later that we isolated the molecule quinine as being the main responsible, or the main bioactive component of that plant. This plant is also one of our leading anti-malarial drugs, right? So it's really good at killing the parasitic infection that is malaria that's transmitted through mosquitoes. However, these compounds don't just have to deal with antibiotics or anti-malarials. Some of these plants also produce compounds that are important to be anti-cancer, right? Such as this compound here, paclitaxel. So nature has inspired and will continue to inspire a lot of our medicines across the board. I mentioned this compound earlier because it really inspired one of the most prominent stories, right? So quinine is an anti-malarial, but during the Vietnam War, there was a large portion of malaria that was resistant to quinine, and the scientist Tu Youyou actually took it upon herself to try to find an alternative compound. And so instead of going through and screening a bunch of synthetic libraries, she turned to traditional Chinese medicine literature and noticed that there was some early description of a plant that was capable of achieving the same effects that the cinchona plant had, right? And that was this plant called the mugwort. And so she spent the rest of her education trying to figure out which compound was responsible. And she ended up isolating this compound artemisinin, and that later turned out to be one of the most effective anti-malarial compounds for those that were resistant to quinine, and saved over 2 million lives and still continues to save lives today. And this discovery was so important that she was actually awarded the 2015 Nobel Prize in Medicine and Physiology. So how do we get from all of those plants and biomass and fungi and mycelium all to an extract of these chemicals, right? Some of you might have seen those keywords that I put out there, distillations and extractions. Here we have a very simplified version of a distillation, right? You can have water up in this flask. You heat it up and it turns into steam, right? It goes into the gas phase. And then it'll go and percolate through your biomass. This is where you would put your mushrooms, your mycelium, your plants, your seeds, whatever. And then it'll go in and extract some of those organic molecules, put them into the gas phase with it, and then you have this condenser that cools that gas down, turns it back into a liquid, and then that liquid can then float down into this receiving flask, right? And if you've ever tried to mix oil and water, you know that they don't mix that well. And that's a similar effect that'll help you separate some of these compounds, right? As it goes into the receiving flask, the water will solubilize itself, and then the oily compounds will separate, and you can then extract them. Another example is solvent extraction or maceration, where you take these biomasses and you crush them up in a mortar and pestle. Or if you've ever cooked and made salsa with a molcajete, it's the same principle, but you're crushing it up to try to break open the cells. Then you'll add some solvent in there, and then you can have what you call an extract, right? And now we're going to be talking about chromatography. How do we have all these extracted compounds in this complex mixture? How can we separate them? Here's an oversimplified example of thin layer chromatography, where you'll have your extract, your solvent, which is your mobile phase, and then your piece of paper, which is your stationary phase. And as you allow, once you stick this piece of paper in there, the paper will start to pull the solvent all the way up. And as it goes up the paper, it'll start to separate your compounds, just like this, right? So you'll have your original extract, and then an example of compound A separating from compound B, and compound C, and compound D. Right, so this is a very simple version of chromatography, but this is a very analogous process that's happening in one of these fancy instruments that we use every day called an HPLC. Looks like this. You've got your solvent that's moving through, and then in this portion compartment right here, you have your column, and it looks and functions similar to this, right? So your solvent's running through, that's your mobile phase. You have your column as your stationary phase, and the whole mixture runs through and separates itself depending on its polarity. It's kind of like a tug of war. Do these molecules like to be stuck on the paper, or do they like to be moving with the solvent? And those different attractions will help you separate them. Instead of looking at spots on a piece of paper, we get this readout that looks like a bunch of peaks, right? And it's cool to see, but how do we go from these peaks to solving that structure? Because I know those peaks don't look like that structure, right? Most of my PhD was kind of spent on that exact thing, right? We'll call that structural elucidation and figuring out the structure. It starts off with your extract, which we just learned how to do, distillation or maceration or solvent extraction. Then we do our chromatography, right, with our TLC spots. And you can take two different routes. You can either look and chase after one peak that you think is interesting, and then you run through different chromatography steps until it's the only peak you see, and then you can characterize it. Or you can go after bioactivity, right? So say if I want to find a new antifungal to fight the zombie fungus cordyceps, then I would be growing that fungus and then putting in those peaks onto that fungus to see whether or not it kills that fungus, right? And if it does, then I continue doing that until I have a pure compound or a pure substance. Then we'll use analytical techniques such as NMR or nuclear magnetic resonance and mass spectrometry that help us decode the chemical makeup and chemical composition. There's also a different technique. It's not as popular, and I definitely did not use this during my PhD, but it's an older technique called crystallography. But before we get into it, those simple basics or fundamentals of crystallography is a well-ordered system. So if you've ever seen your table salt, it looks like tiny little crystals, and if you zoom in on a microscope, I'm almost certain they'll be square. Those crystals exist with other compounds that aren't sodium and chloride, like your table salt. Here's an example of a crystal of proteins, right? And if you zoom in even further using a transition electron microscope, you can start to see how well ordered these are. They're in a straight line. Some of these might be positioned differently throughout the sequence, right? Adding some imperfection to that order, but we can still utilize this. We utilize these, or we can visualize that image through a microscope called the transition electron microscope. But before we go into it even further, just to give you a relative scale or sense of the magnification power of this microscope, here we have an optical microscope that most of you might have seen in your classrooms before, and visualizing blood cells, right? That same blood sample can be visualized in a higher definition using a scanning electron microscope, right? So here we're bombarding these samples with electrons, and they're bouncing off, and we can get a very high resolution image. Some of you might have seen this image before on the internet or something similar, right? This is the face of an ant. And we can have similar images that describe other insects, like praying mantises, cockroaches, or the tardigrades. But all of those details and magnification power is thanks to the scanning electron microscope. The instrument that I use on a regular basis is that of a transition electron microscope, right? So here we have electrons bouncing off of these samples. The transmission part means it goes through it. So these samples need to be thin enough for those electrons to go through. And because it's so powerful, you can magnify even down into this level. We can also use this microscope not just to image, but to collect diffraction data that you'll see here. But before we get into it, how do we go from that diffraction into an atomic model like the one we see up here? The basic schematic of this is you'll have these X-rays or particles. In my case, I'm using electrons, right? And I'll tell you why in a moment. You have these particles that go through this well-ordered system, and because it's so well-ordered, it has a repeating diffraction pattern, right? And the more repetitive it is, the higher the accuracy that happens. Then some fancy math that Ms. Gilbert will probably understand, called the Fourier transformation, can take that diffraction pattern and produce an electron density map. And from that density map, you can then derive your atomic model. And that's essentially how we can take a crystal and bombard it and get ourselves a three-dimensional structure. So why use electrons over X-rays? The main reason is because in nature, right, a lot of these compounds are very complex, and they can only form really small crystals, right? Things that are almost invisible to the eye, most of the time. But electron and X-rays can't penetrate things that are too small, right, such as this. And so if we're shooting X-rays at these tiny crystals, we might not get as high of a diffraction rate, and therefore we won't get data that will help us solve the structure. But electrons travel at a smaller wavelength. They have charge and mass, so it makes them more likely to interact with that crystal and have those diffraction patterns, and therefore electrons are a good source to have this diffraction on these small crystals that come from nature. And so this is a video that kind of describes the process of having these tiny crystals and how we prepare the samples, right? So once you've grown your crystals, you can plunge freeze them and place them on a grid here. And now it's going to be inserted into that transition electron microscope that I showed you, right? And as it's in the microscope, there's a beam that comes down and transmits through there and causes this diffraction pattern. And we can collect these individual frames that we can then use to do fancy math, the Fourier transformation, and get some electron density maps. And so our group was probably leading the field here in applying that same technique to small molecules, right? And there was a former group member named Li Jun who was one of the first to apply this to natural products. But the original workflow kind of went from extract to individually isolated and purified molecules, right? They went through this process iteratively, got one sample. Then they took that one individual sample, placed it on a grid. You can see these crystals, or depiction of crystals there, put it into the microscope. This is what a real image looks like of what we would call a drop cast. And all these tiny little dark particles here may or may not be crystals. Or ordered crystals. And you go through, right, you locate a nice little crystal, and these are definitely invisible to your eye. There's no way you would be able to see these unless you've got supervision. And once you find those crystals, you can go ahead and collect some diffraction data, right? And this is a really nice process, and Li Jun really showcased the power of the application of electron diffraction to natural products. And that's when I really got excited, and that was around the time that I started here in the group. They were able to solve the molecule lomivitacin C, which is super complex, and if I was to be tasked to do this in graduate school, it probably would take me several years to do that, but she was able to do it in a couple months. So that's where I came in, and I told Hosea, I was like, I want to be able to do this, but on a discovery scale, right? Instead of having to go through and purify all these samples and spend all that time doing iterative purification, I want to see if we can just take things straight from nature, put it on a grid, and see if there's any crystals on there, right? Skip all the steps, save people time. Although the instrument's expensive, it should save you some money in the long run. And so if we take this schematic that I showed earlier, where you have single compounds being drop casted onto a single grid to get diffraction, to get a single structure, my idea was to take this whole plate, not just one sample, and utilize this instrument called a microarrayer to drop tiny little bits of each sample into a nice uniformed array on these grids. And so that's essentially what we set out to do. And our first case basis came from a fungal extract, right? Here we have 49 milligrams of hypoxylon. We ran it through chromatography, and we were looking at these peaks, right? And then this blue region is all the different fractions, right? So there's 96 different lines. It's hard to see on this projection, but we wanted to go ahead and label or dispense all those 96 different fractions onto this grid. And so you can see these tiny, tiny little drops being dispensed in a nice ordered fashion until you get all 96 samples on there. And since I like to believe that seeing is believing, instead of just seeing that, this is what an actual microarray looks like, right? So this is on a super magnified scale. You can see all these tiny little droplets, and if you zoom in even further, our hypothesis actually turned out to be true. There are these tiny microcrystals that can exist in nature that we normally don't see using other microscopes or other diffraction techniques. And so here you can see even just the sheer scale of how small these crystals are. And these are the types of crystals that we're utilizing every day. And so in this case basis, right, we took this extract, arrayed it on there, we found those tiny little crystals, some like these, right, collected some diffraction data readily, and then collected a rotational data set, right? So we can't just get these still images, you have to rotate the sample around. And you get a nice little movie like this. And even though I didn't become an astronaut like I wanted to, I still look at this image every day and I'm just like, this is similar to the stars that are in the sky. And so it makes me happy to look at these guys. Through this process, right, we've been able to characterize several different natural products without having to spend months to years characterizing each individual one. And since I wanted to discover new things, right, in the short 2 years that we've been at it, we've been able to discover 9 new natural products, including a whole new class that's never been seen before or never been published before. And by continuing to do that, we've also been able to characterize tons of new or known natural products. But since this was discovering medicines, right, we wanted to kind of combat some antifungal diseases. And so one of the compounds that we actually characterized, although it was known, we wanted to kind of find some antifungals. We tested all of the new ones, but turns out they're not that great at killing fungus. But one of the compounds that we isolated along with those new ones is pretty good at killing fungus, and it also has a new structural scaffold, right? There's no tetramic acid functionality that's been known to kill fungus, and so maybe we might be introducing a new scaffold that other scientists might be able to use. And so all of this definitely wouldn't have been possible without the group supporting me. We've been able to go through and screen almost hundreds of extracts and characterize several tens of molecules. And I wouldn't be able to do that by myself. And I definitely wouldn't have been able to carry out this workflow alone. So the people in red definitely helped me in optimizing that workflow and carrying it out. And I also wouldn't be here without fellowships that supported me throughout my career, such as the Louis Stokes Camp Fellowship from NSF, the UC Leeds Fellowship that helped me get the summer research internship experience that really propelled my interest in research. And definitely wouldn't be here without my mentors, especially Hosea, who gave me the freedom to do a really expensive project in his lab. That was a lot of risk with some reward. We didn't think it was going to be possible to do this, but turns out it was. And so without him giving me the opportunity, I wouldn't be here speaking to you today. And then, again, I want to acknowledge all of the people that were in my mycelial network, right? My mom, dad, Ms. Gilbert, Ryan Baxter, Peter Mai, Lizzie, Daniel Suez, Troy Stitch, all of those guys, Hosea. And this is just to show that, you know, most of this wouldn't have been possible without one supportive or caring teacher. There was a lot of indecision in my life at that moment, and I think Ms. Gilbert caught me right at the right time and pretty much set all of this into motion. And so for that, I thank you. And then as you leave here, right, most of the discoveries, everybody wants to say, you've got to come up with an idea first, and that's the only way things will be discovered. But I like this quote by Ernest Schrödinger. The task is not to see what's never been seen before, but to think what has never been thought about what you see every day. So when you guys leave this auditorium today, just look around, and instead of looking at grass as grass, or plants as plants, maybe you can start to look at them as molecules and their potential that they might have. And so, yeah, with that, that's all I got.
All right. Thanks, David. That was amazing. All right, so we are going to have a short question and answer session. So if you have questions, please come up to one of the mics in the aisles, and we'll take you one at a time, obviously. And David's really excited to see what you guys want to know. All right, first question over here.
I know what the thing is, like micrometers, but what is 3? With the exponent 3?
Yeah, so that's microns cubed, right? So then you're thinking about it in 3 different dimensions. Yeah.
So it's like 3, how many times micron is that?
So you're thinking about volume, right? So the crystal could go 1 micron in this direction, 1 micron in this direction, and then 1 micron in this direction.
So for example, 2 by 2, 2 microns cubed would be 4 microns in volume.
No. 2 by 2 by 2.
Oh,
Yeah.
okay. Bye.
All right, over here.
What is the best life advice you could give to anyone?
Best life advice is always hear people out. Right? Always have an open ear, I would say. I know a lot of us have an idea of what we want our life to be like or how we want our day to go, and we know that doesn't always happen. Right? But there's always people that have gone through similar things that you're going through or have gone through the careers that you want to go through, and so just ask and seek for their advice and hear them out. Right? Listen to what they have to say. And whatever wisdom they might impart on you. Yeah.
All right, question here.
Hi, how do you take the molecular structure when you find it in a plant and then turn it into medicine?
Yeah, so that's a very good question. So a lot of the work that we do when we're looking for drug leads is we'll associate that compound or that structure with the activity, right? We'll say this thing is anti-cancer, antifungal, but then it has to go through several different clinical trials, right? And most of the time, the things that we find in nature will fail, right? They're not selective. They won't only kill the cancer cells, they also kill your normal cells, right? And so that's where these medicinal chemists will come in and try to change little portions of that molecule until they get that selectivity, right? So it only kills that cancerous cell versus your normal cells. And then they'll start messing around with other properties, but eventually they'll have to go through all those clinical trials to become a medicine. Yeah.
All right, over in this aisle.
Does your current career involve you studying space?
I don't know if I could say it, but you can use this technique on most samples, right? So you can go out and grab dirt thrown on a grid and try to diffract on it. You can think about doing that with stuff from Mars or space, right? Or from a comet to figure out what the molecular makeup is of those samples. And so I haven't done it yet, but that would be super interesting to do. Yeah.
All right, next on this aisle.
Can you guys see atoms?
Yeah. Well, we see what we call the shadow, almost like the shadow, right? The electron density map. But through this technique, it is probably, most of the crystallography techniques are the best ways to visualize those atoms in 3 dimensions. So, yeah.
So my question is it true that mushrooms can make you dizzy and make you get confused? Is it true?
Sorry, can you repeat the question?
Is it true that mushrooms, if you eat it by itself, then would you get dizzy?
Yeah. There's definitely mushrooms out there that are toxic to us, right? And they'll have those effects where they'll make you sick. One of those is like the Amanita mushroom from, Alice in Wonderland, right? That famous red one. Those will make you really sick and nauseous. So yeah, definitely don't go out and pick mushrooms and eat them.
Yeah, but is our immune system strong enough to defend it?
Is our system strong enough to defend? I think it depends on how much you eat. Yeah.
Okay.
So don't eat too much.
All right, I think we'll do one more from this one because she's been waiting a little bit, and then we'll come over to the aisle over here.
My question is that whenever observing really small experiments or anything, do you ever consider anything about the observer's effect?
The observer's effects? Yeah, no, that's actually a brilliant question, and definitely something that takes into account. So the scanning electron microscope and the transmission electron microscope, although it gives us the benefit of increased magnification, right? We can see into a higher field. It does come at a cost, right? We're bombarding these samples with electrons, and therefore we're changing what they look like. And so some of these samples, if you put it in a scanning electron microscope, if they've got water in there, and you're shooting it with electrons, you can start to see them get charged, and they'll start to glow and peel over and move. And the same thing happens with some of the crystals, right? We call it beam-induced emotion. And so that'll definitely affect the data quality that you collect. But yeah, great question.
All right, over on this aisle.
Are you still interested in space, or are you done with that?
I don't think I'll ever be done with space. I think I like to be on the edge of discovery, and there's lots of things left to discover in space, right? There's even things in the bottom of the ocean that I'd want to explore, but I don't think I'll ever be done with space. Yeah.
All right, we're going to take 2 more questions, so we'll come for this one, and then we'll do that one last.
So for the fungus that got into the brains of bugs, is it what's preventing it from not taking over humans, basically?
Yeah, that's a great question. So I'm not exactly sure, but my intuition would be the temperature of our body, right? So what stops us as mammals from getting a lot of fungal infections is that these organisms such as fungus can't exist above our body temperature, right? So that will help us defend against a fungal infection along with the immune system, right? So it comes together from multiple sides. But I think for the most part, it's definitely our body temperature, yeah. But that's why a lot of people are talking about anti-fungals, because climate change is changing the temperatures at which these organisms are existing. And if that continues to rise, these organisms are going to evolve to be compatible at temperatures like the ones found in our body. So we got to get ahead of the curve there.
That's cool. All right, last question, and then we're going to get to our raffle.
When you were talking about the aloe vera, right before that, how did it help with the headaches and the fevers and stuff?
Yeah, so I would say headaches, it was more anti-inflammatory, right? So if you think about aspirin, like the molecule that I showed that came from the willow tree, aloe vera also has that same molecule in there. It's just not as abundant. There isn't as much of that compound in aloe vera as there is in the bark of the willow tree. So it does help in that regard, but just not as well. Yeah.
All right, those were great questions. Thank you, everybody, for asking. And sorry to those of you who didn't, but hopefully you can look David up and ask him personally if you want to.